Good day, guys. So in this video, we are going to be learning about DNA code of life. So when we look at our guideline, it tells us that we need to know what is a cell. Okay, so it is very important to know the background knowledge. Okay, so this is the structure of a cell, but what is a cell basically? So a cell is the basic unit of life. Guys, in life sciences, you need to be curious. Each and every time when you read something, you need to ask yourself, what is that? And why is it like that? Okay. So a cell is the basic unit of life. It's like a tiny factory that does everything from breathing to thinking. All living things are made up of them. All living things are made up of what you see right here. Okay, which is a cell. So every living thing has it. So let's try and understand terminologies, which is very important when it comes to life sciences. If you don't know these terminologies, then forget. You are not going to know anything. It is very important for you to know these terminologies. So what is the nucleolus, right? So it is a structure in the nucleus responsible for forming ribosomal DNA, right? And then we have a nucleoplasm, the part of the protoplasm within the nucleus. We have cytoplasm, the part of the protoplasm outside the nucleus. Ribosome structure, that is the site of protein synthesis. Okay, so it is basically a site where protein synthesis happens, which we are going to get into what is protein synthesis as time goes on. Okay, so now let's come to the important ones, the ones that you are going to see a lot. Okay, so we have chromatin, right? So a chromatin basically, it is the DNA containing network found in cells. Okay, so it is the DNA containing network found in phase, in interphase, non-divided, right? And then we have a chromatid. So make sure that you understand the difference between these words because they sound very similar. Okay, they sound very similar. So this one is a chromatid, that one is a chromatin. So it is the chromatid, the individual threads that form on a chromosome. A centromere, they say it is a structure that holds two chromatids together, right? So basically, we have a chromatid right here. This is going to be our centromere. And then we have another chromatid, right? So you can see there, we have these. That is the centromere. It is the structure that holds two chromatids together. So this is a chromatid, and then this is a chromatid. Okay, and then we have a chromosome. A chromosome is a thread-like structure made up of DNA that carries hereditary information in the form of genes. A chromatin network, it is a visible as thread-like structure in the nucleus of an individual cell. So you have your nucleus right there. Your chromatin network would look something like this. Okay, so it would look something like that, where you have a lot of threads and a lot of threads forming together. So it would look something like that. That is a chromatin network. Okay, right. DNA, which is known as deoxyribonucleic acid. That's what DNA stands for, deoxyribonucleic acid. It forms the chromosomes in the nucleus of all living cells. We spoke about what is a chromosome, so DNA forms it, okay, in the nucleus. So we also spoke about the nucleus of all living cells and carries hereditary information, right? A DNA molecule is double helix, twisted strand, very important. This is very important for you to know. It is a double helix strand. RNA, it is a single strand. It is located in the nucleoplasm, and also something that you need to understand. It is always a single strand of nucleotides, okay? A helix is a cold natural shape of DNA. So basically when they say a DNA is helix, that's what they're talking about. It is a cold natural shape of a DNA molecule. So a monomer, right, is going to be a single unit that makes up a larger molecule. What is a larger molecule? A molecule is two or more atoms stuck together. Therefore, a monomer is going to be a lot of two or more atoms stuck together. That's what basically they mean. And then a polymer is a large molecule. Okay, so this one is a single unit that makes up a large molecule. but this one is a large molecule which is formed from many small molecules which are monomers, right? So a polymer basically is made up of monomer. So a monomer are more like a lot of atoms, right, stuck together, which is a molecule. A polymer is a monomer, right? A polymer are a large molecule of monomers, basically. So you need to understand, guys, ne? So a monomer is made up of a lot of atoms, but a polymer is made up of a lot of monomers. So that's what it means, basically. Okay. Let's come to a nucleotide. A nucleotide is the building block, right? Monomers of RNA and DNA. So nucleotide, polymer are made of the same thing. They are building block which are monomers of RNA and DNA. So RNA and DNA. Each nucleotide consists of a pentose sugar. What is a pentose sugar? Basically, a pentose sugar is a 5-carbon sugar like ribose or deoxyribose. So basically, it is the backbone of DNA and RNA. Okay, so you need to understand it is a 5-carbon sugar like ribose or deoxyribose. That is a pentose sugar. Let's go to amino acid. An amino acid is the basic building block monomer of a protein molecule. So very similar to each other, but they are different at the same time. Okay, so an enzyme, it is a protein that speeds up a chemical reaction, right? So an enzyme is a protein that speeds up a chemical reaction. So for example, where you are making your tea, right? You are making your tea and you want your sugar to dissolve in your water. So if you want that to happen much more faster than it already happens, then you are going to use a catalyst for that to happen. Okay, so we have our pairs right here. We have our 3 pairs, okay? We have cytosine, which is the base that pairs off with guanine. We have thymine, the base that pairs off with adenine. And then we have uracil, the base found in RNA and not DNA. So I want you to catch these things very simple. So we have guanine here. Guanine starts with a G, okay? So this G, you can say it is garage. What goes into the garage? What goes into the garage is a car, right? A car goes into the garage. Therefore, G pairs with C, as you can see right here. G pairs with C. Car going into the garage. Now I want you to take your T. This is your tree. Okay, what falls from trees? Apples. Okay, so therefore apples falls from trees, therefore T goes with A. That's how I want you to understand this. Okay, but this happens in DNA, right? It happens where? In DNA. In RNA we do not have thymine, right? In RNA we do not have thymine, therefore this only happens in DNA, and because we don't have thymine in RNA, right? So the base found in RNA, so we have uracil that is found in RNA. So therefore, U, that's going to pair with A. In DNA, we have T that pairs with A. Okay, so very different. They don't go together in each and every situation. You need to look in which situation you are dealing with. Okay, right. They are called nitrogenous bases, by the way. So base pairing, adenine always bonds with T, which is thymine, apples, cheese, guanine with cytosine, car, garage, in DNA molecule, okay? And then we have now the locations of DNA. We have DNA located in the mitochondria, which is known as mitochondrial DNA. We have DNA located in the nucleus, which is known as nuclear DNA. Chloroplast DNA, this one only happens in plants, found in plants. That one is only found in plants, okay? Now let's talk about transcription and translation. Transcription is the first stage of protein synthesis. Most students tend to mix the two. They think that translation happens first and then it's transcription. No, transcription happens first. As you can see here, it is the first stage of protein synthesis. And then translation, which is the second stage of protein synthesis, happens after transcription. Okay. Protein synthesis is a process where cells read DNA to build protein. Okay, we spoke about protein. So it is a process where cells read DNA to build protein, which is mRNA, which will go to your ribosome. Okay, so basically that's what protein synthesis is. And then synthesis stands for building up separate parts in a whole. Basically, when we take something and we are going to build a whole thing with it, okay? It's like when you take your sugar, right? And then when you take your water, you are taking separate parts of something and then you are going to turn it into a whole, which is your tea or your coffee, okay? So we have messenger RNA, which is known as mRNA, which is responsible for carrying the genetic code. Did we speak about genetic code? No. So genetic code is the setup of 3 letters in DNA or RNA. Okay. So those 3 letters are the ones that are going to go to the ribosome where the information is translated to protein synthesis. So it carries code, as you can see, genetic code. That genetic code is the set of 3 letters in DNA or RNA. Okay. And then we have ribosomal RNA. Ribosomal RNA form the ribosomes and produce proteins based on the information received from the tRNA. So ribosomal RNA forms the ribosome and produce proteins based on the information received from the tRNA. What is tRNA? This is tRNA, right? So it has anticodons. What are anticodons? We are going to talk about in the next page. It has anticodons which code for a specific amino acid. The anticodons are complementary to mRNA. What do they mean when they say they are complementary? You and your friend, you'll go together, but you are not the same. You don't look the same, okay? Therefore, that's what they mean when they say they are complementary to RNA. They go together, but they are not the same. They don't look the same. A codon is the 3 adjacent bases found on a mRNA molecule. Okay, we spoke about what is a molecule. So a codon are the 3 adjacent bases found on mRNA, and then anticodon are the 3 adjacent bases found on tRNA. Codon, mRNA, anticodon, tRNA. Okay, so don't confuse the two. Hydrogen bonds, right? So it is a chemical bond which links base pairs in DNA molecule. Remember that hydrogen bonds, we find them in DNA molecule, right? Peptide bond, it is a link between 2 adjacent amino acid. A peptide bond, take it like a glue, guys. Take it like a glue, as well as hydrogen bonds, but that they have different functions. A peptide bond is found between 2 adjacent amino acids, okay? Right, so a gene, it is a segment of chromosome. We spoke about what is the chromosome that controls each characteristic, a unit of sequence pieces of DNA that carries the genetic information that will determine the hereditary branches of a organism. And then, hello, matriculant. Did you know that Mobile Education offers live online tutoring where you can ask questions to the tutor and the tutor will answer you immediately. We have 3 plans. We have the basic, standard, and premium plan. The basic is 120, standard 200 rand, and premium is 250. You can pause the video and look at the benefits each plan has. Thank you very much. Enjoy the rest of your video. There we go. This word, we've been reading about it, but we didn't know what it was, okay? So it is characteristics that are passed from parents to offspring, right? It's like you having a big head, and then your mother also has the big head, right? That's basically it. Those characteristics were passed on from you, from your mother to you, right? So a mutation is a mistake, guys. A mutation is a what? A mutation is a mistake. It's not something that's supposed to happen, but it happens. And then here they define it as a sudden change in the DNA nucleotide sequence. Okay, so it is a sudden change in the nucleotide sequence. Hello, matriculant. Did you know that Mobile Education offers live online tutoring where you can ask questions to the tutor and the tutor will answer you immediately? We have 3 plans. We have the basic, standard, and premium plan. The basic is 120, standard 200 rand, and premium is 250. You can pause the video and look at the benefits each plan has. Thank you very much. Enjoy the rest of your video.